Good morning. This is Dr. Garais, MED110, Anatomy and Physiology, and it is now week 4, Wednesday, November 4th, 2020. And what is due today? Task 3, discussion 3, lesson 3 is due today. I'll give everyone by closing of business. I don't know, 6, 8 o'clock today was when I start looking at grades. Next week, we are now in, of course, week 4. Next week is your midterm. What's due for next week? And I'll discuss midterm protocols in a minute. What's due for next week? Of course, your concept map for task 4. Now, I'm going to cut you guys some slack for discussion 4. Okay. Ooh, some people did it. Nice. Let me put here. Task. Discussion 4 is for extra credit. If you decide to perform discussion 4, then it will be applied to 10% of your midterm exam grade. Okay, let me post that up. So discussion 4 is optional, and I see some people already, out of habit, already performed it, which is good. And 10% is always good. And lesson 4 would be, of course, this application assignment down here, which would be mitosis exposed. Is it's kind of corny, but it's got some things about. Let's see where the questions are. Right, so it gives a little story, like they make it like a courtroom analogy on mitosis versus meiosis, which is what we're going to do. And there's only 5 questions at the end of that. So next week, the only thing that's due is task 4 and these 5 questions at the end of the case in lesson 4. Discussion 4 is optional. Repeat, discussion 4 is optional. Okay, now how's the midterm going to go down for next week? Next week, when you at 9 a.m., well, 5 or 10 minutes before 9 a.m., you're going to look in here. You don't need to do Zoom. You're going to look in here. And then there's going to say, lecture something or other, right? Not lecture, midterm, the actual midterm. I'm going to put a, what do you call that? A practice midterm later this afternoon for you guys. And remember, that's just practice. Don't ask me for a key or the right answers. That's your job. Just use the practice midterm. It's gonna look similar to your final, but it's not gonna be the same thing, but it's gonna be the same format. 50 items, multiple choice. It's gonna have some pictures in it because it is an anatomy class. Sorry. I gotta share the screen properly. Okay, can you see my screen now?
Yes.
Can you see my screen now? Either unmute or put it in the comments.
Yes.
Yes, thank you.
We good?
Yes.
Hello? Hello? Does everyone see my screen now?
Yes. I can see it.
Yes, good. Okay, So, of course, it's going to, next week, a couple of minutes before 9 a.m. The exam is from 9 to 10:15 a.m. So this is what's going to happen. You're going to have, your exam. It'll be in a Microsoft Word document. So at a couple of minutes before you log into your Moodle, a couple of minutes before 9 a.m. next week, and then there'll be no lecture. I'll have a video lecture in its place, so that we don't miss out on lecture. And um, uh, I think we also have to check if we have laboratory. Let me see, but I'm sure I didn't put the laboratory dates. No, we'll talk about that next week. But next week is the midterm 50 items multiple choice. I'm going to give you a sample of what that's going to look like. So when you come here, let's pretend that it's next week 5. A couple of minutes before, like 8:50 a.m., you go in here and it'll say midterm. You open up the Microsoft Word document and there should be a midterm. Now, and then you just send me the answers via email, and I'll have my email address as well within the message or within the announcement. That's all I need. I need numbers 1 through 50, your full name, and just the letter answer. That's it. Don't resend me the exam with highlighted answers because it takes too long to grade and it's too complicated. It's just quicker when I just have the answers, okay? And remember, there's going to be a practice exam that I'm going to send, and then next week at 8:50 a.m. there's going to be the actual exam, okay? So that's what we're going to be doing next week. And at the end of this lecture, I'm going to be showing you how to study for a multiple choice examination. If you're not familiar with how multiple choice examination strategies, I'll also be discussing strategies on how to study. Do not pull an all-nighter. I'll get into that. Don't try to cram information because it will not bode well for you, not for this course and not for any of your future courses. Okay, so let's start with today's lecture proper, and then we'll now talk about the exam strategies and the sample just some sample strategies and sample questions. So for this week, the scope of the exam is everything that was ever discussed, including cases, up to and including week 4. Okay, so what are we talking about this week? We're talking about cellular biology, and then we're talking about DNA, deoxyribonucleic acid. And of course, in that same vein, we're going to be talking about RNA and protein synthesis, how do cells grow and divide, and how we already kind of know how we tell cells apart. And I'll remind you guys about that within the lecture. Yeah, Miss Newman, I saw it. I will be I saw your items. I will send you that later, right? And thank you for, again, if I discuss something with you, just in general, and he goes, communication is key, and thank you for reminding me if I didn't get back to you like really, really quick. Quick as in less than 24 hours. Okay, so these are our 4 topics: structure of the structure and function of DNA, DNA replication, mitosis, and meiosis. And let's talk about nucleus and structure of DNA. And if you go here into the lesson, oh, and by the way, I mentioned earlier, the discussion 4 is optional. And if you did it, you already now have extra credit that will be applied to your midterm exam grade. And I wrote a little message in there in discussion 4. So let's go to lesson 4, and there's this lovely video right here. This video is golden, and We're going to watch it together, and then I'm going to write some notes here on the side. And again, will be a nice little lesson on how to take notes and how to know what's important. So right now, we now know that we have to know as much as we can regarding DNA. And there's there's this lovely video here. So we're going to watch it in sections. And I wonder why it's muted. And I can't hear it. Well, it's odd. Well, it doesn't matter. This is what I'm going to do. I'm going to just talk over it and show you some pertinent pictures. So what this guy, I forgot his name, Alton or something or other. What he is talking about is the DNA structure. Now, the DNA structure is a double helix. And so it has twists and turns, and that's what a helix is. It's like a corkscrew. Let me see if we have a picture here. So we have two things. DNA, deoxyribonucleic acid, and RNA. The DNA, both of them are made up of pieces. Let's see if we can bring the video to. Okay. So it's kind of like Lego. You have the pieces that put together, and when they're all glued together, they make this chain, and there's two sides to this chain, right? But first, let's talk about the parts. So you'll hear the word monomer versus polymer. A monomer is just like one piece of part or one piece of Lego to make one big chain, which is a polymer, right? So mono means one, poly means many. So when you think about a chain, it's made up of a whole bunch of links. And DNA is made up of a 5-carbon sugar molecule, right? Which is ribose. How do I know this is 5? 1, 2, 3, 4, 5. It's a pentagram, right? So if you haven't had your biochemistry yet, each corner is a carbon molecule. And this 5-carbon sugar is ribose. Then you have a phosphate group, and then it attaches onto one of 4 nitrogenous bases, like randomly. And the nitrogenous bases are A for adenine, C for cytosine, T for thymine, and G for guanine, which they're going to show in a minute. So right off the bat, doesn't this look like a beautiful all of the above question? Which of the following are pieces or parts of a DNA monomer? Is it ribose, a phosphate group, and any one of these nitrogenous bases? And they're called nitrogenous bases because they're basic or base, right, versus an acid. That means they have an OH minus group somewhere on here, and they also have a nitrogen on there. And they form like the chain or the rungs of a ladder of DNA. So let's look at bases. That's deoxyribose. Okay. So this is what a typical DNA chain looks like. It looks like a ladder. So this is a rung, the steps on a ladder. So you have your phosphate backbone, which is like, the edges of the ladder. Then you have the 5-carbon sugar, which is the ribose here. And if you notice that the ribose, the oxygen group right here, they're pointing in a different direction. Okay, so this is called 5' to 3' and 3' to 5'. So one side of the ladder is going up one direction, and the other side of the ladder is going up another direction. How do I know it's DNA? Because there's two sides, right? And this, because of the hydrogen bonds in between the adenine and thymine and cytosine and guanine, which by the way, any adenine has to hook up with thymine. Any thymine can only hook up with adenine. Any guanine can only hook up with cytosine. Any cytosine can only hook up with guanine. And because of the hydrogen bonds that are in between them, they're soft. That means it can be compressed and it twists. So this ladder forms what they call a double helix, or a twisting double-sided structure. And you could see here, this is one side that's going in one direction, and there's another side going in another direction. How do you know what direction they're going into? If you look at the oxygen here on the ribose, so that you know that one side's going up that way and this side's going up the other way, okay? And this side's typically called the 5' to 3' because it's a biochemistry thing. Just know it's one direction, and then the other one is another direction. So again, what questions could I ask you here? Could I give you a picture of this, right? And then I could ask you to identify what this is. Oh, that's a nitrogenous base, adenine. What's this? That's a thymine base. This, cytosine base, guanine base. This, 5-carbon sugar, ribose. And this is a phosphate group forming the backbone of this DNA molecule. And DNA, we've mentioned before, these ATCGs form like a code. So DNA has the code or the master plan or the blueprint of everything that you are. And I'm not exaggerating when I say everything, everything. What color your eye is going to be? Are you going to be an alcoholic? Are you going to have major depressive disorder? Are you going to have anger issues? Are you going to have daddy issues? All of this, actually, behaviors are 40% programmed into this. This code, in theory, knows when you're going to be born, how you're going to be born, when you're going to die, how you're going to die, what diseases are you predisposed to, way before you even are introduced into the world. This should amaze you, because all of that information, which is billions and billions of lines of code, is all scrunched into something smaller than a cell. And we all remember that the nucleolus has a compact DNA, and in the rest of the DNA, you have chromatin. And this code is the expression of all of that. So let's look at, so that's why they call it 5' and 3'. It's the directions at which it's where the phosphate group's connected. But just know that one side's going one direction, and the best way to look at it is where the oxygen is pointing. So wherever the oxygen is pointing, that's 5' to 3'. And then if it's pointing down the other way, that's 3' to 5'. And that's how you know it's DNA. Another reason why you know it's DNA, because there's adenine, thymine, cytosine, and guanine. Now, let's now look at the base pair matchups. I stated earlier that adenine only can match up with thymine, guanine can only match up with cytosine because of the way the hydrogen bonds are formed. So if we look at this, right? Adenine and thymine have 2 hydrogen bonds. Guanine and cytosine have 3 hydrogen bonds. This is important because this helps form the twist of the DNA molecule. Because if you look overall of what a DNA molecule looks like, it looks like this. And that's why they call it a double helix. It looks like a staircase or a ladder that twists and goes round and round and round. And it's because of the double bonds here. And as you can see, and not double bonds, the hydrogen bonds. As you can see, for adenine and thymine, there's only 2. Guanine and cytosine, there's 3. That's a good question. That should come out. Okay? And that's what makes it twist. And also, the other reason why whatever or whoever created us, and you can see this is highly engineered, it forms a code. So let's look at the code. So He gave an example here. AGGTCCATG. Let me write that down, and then I'm gonna show it on a. I'm gonna show it on Microsoft Word, because this is a potential question on an exam. So, and also, this is a classic NCLEX question too. So if, let's say I have DNA, and I told you that one side will be the 5' to 3' side. Okay. So let's say the code was A, G, G, T, C, C, A, T, G. Say that's the code. And it's going in one direction. So we have to know that the other side of the DNA chain, so this is one side of the DNA chain, the other side has to be 3' to 5'. And we know what matches up with who. So the A, adenine, always has to match up with thymine. Guanine always matches up with cytosine. Thymine always matches up with adenine. Cytosine always matches up with guanine. Cytosine always matches up with guanine. Adenine always matches up with thymine. Let's move this a little bit further so they match. Thymine always matches with adenine, and guanine always matches with cytosine. So if this part, okay, the 5' to 3', that's called the leading strand. The 3' to 5' is the opposite side. That's called the lagging strand, and we'll talk to you about why. So if I give you the leading strand, you should be able to get me the lagging strand, okay? Because we all know that A's always goes with T's, and T's always go with A's. G's always go with C's, and C's always go with G's, okay? So I could have an exam. If given the 5' to 3' DNA leading strand, which of the following would be its corresponding lagging strand? Or can I give you a lagging strand going the opposite direction and then you giving me the leading strand? Yep, you could. And this is a classic DNA code question. And don't be surprised if you see something like this on your future NCLEX. And that's why I was trying to what I was always saying. Just because you have this class doesn't mean that this information goes away. It'll always come back. Yeah, apples on table and car and garage. Whichever way you acronyms are really neat. How to remember stuff, like I forgot already. The little saying that you have for. There's a whole bunch of them in medical school. One classic acronym that I always still remember to this day is what are the 4 muscles of the rotator cuff? And it sits: supraspinatus, infraspinatus, teres minor, and what's the other one? Subscapular, right? Whatever you need to do to remember it, ATGC. For whatever mean, I always say ATGUG. I don't know. But again, memory is a funny thing. Whichever way you remember it, or what we showed earlier visually. Okay. So do you think now that I spent some time doing this, you could look up questions like this or make up questions like this, and also expect 1 or 2 questions like this on the exam? Hint, hint, hint. Okay. So let's see if we were right. So if AGGTCCATG was one code, TTCCAGATC, let's see if that's correct. Well, TCCAT, yeah, looks the same. Oh, why TT? Or maybe I copied it wrong. Oh, I may have copied it wrong. But that also brings a nice little little question. Like with all these codes, do you think that sometimes the DNA will make mistakes? Well, there's proofreading enzymes, and we're going to talk about that momentarily, that go back to make sure there are no mistakes. And there are some mistakes in the code that don't lead to anything fatal, but there are other mistakes in the code that could lead to something very, very serious. So let's go back. Let's now go to, he just talks about here in this video, oh, AGGGTCC. See how it has one 5' to 3', that's leading. And then there'll be, oh no, so we were correct. TCCDAGGC, and it's going in the other direction. Okay. Let's now get to RNA. Now, what's RNA? Right? DNA is deoxyribo. D means no or not. Well, we have a phosphate group, and then we have the ribose, but looky here. This is oxygen, and that's the difference between DNA and RNA. And RNA is only single-stranded. It's not double-stranded. So D for double-stranded DNA. RNA is single-stranded. DNA is the blueprint of who you are, and RNA is kind of like the messenger or foreman who translates that code and then brings that code, if you recall, remember the rough endoplasmic reticulum in last week's lecture and the ribosomes? Those are the protein factories. Since the code is in a specific order, that code codes for amino acids, and it will create proteins. And we're gonna show you that visually in a minute. But do you think I could also ask the difference between DNA and RNA? If I show you a single strand of something going in only one direction, and this is a 5' to 3' direction, and how do I know? The oxygen, this oxygen here on the ribose sugar is facing upward, and I know this is RNA because it's only single stranded, and there's this huge oxygen right here. Okay. And the function is to take the RNA takes that code out of the nucleus and into the rough endoplasmic reticulum or into the ribosome. So the ribosome has now instructions on how to make a specific protein. And we already know that proteins they're very, very important in our body. They form enzymes, they form vitamins, they form a whole bunch of things. Now, the difference between RNA is their bases. Same phosphate backbone, same ribose sugar, but with the oxygen kicker here. But if you see here, it has adenine, it has cytosine, but instead of thiamine, it has uracil. Oopsies. And then there has guanine. So could I ask you the differences between RNA and DNA in its function, in its appearance? Sure. And the RNA is single-stranded, not double-stranded. The RNA has uracil, not thiamine, because it has to translate a different code to the ribosomes. Does the DNA go outside the cell? No, it does not. Outside the nucleus, it does not, but the RNA does. And you could see here how, if this is the messenger, do you think I could mess up the message like in HIV? HIV latches onto this RNA, changes the message, and eventually also changes the DNA code as well. So that instead of the message to the ribosomes, hey, make some proteins, it'll say, hey, make some viruses instead. And that's why viruses are so ubiquitous. They're very small, they get in, they don't have its own DNA or RNA. It is a parasite and it latches onto yours. That's what makes them very dangerous and hard to kill. So those are the differences between DNA and RNA. And we now also know the difference between the function of DNA and RNA. Now he starts talking about Watson and Crick. That's for your discussion. Let's get to something cool. And he reiterates, okay, how does this thing how does the DNA, and I know this is DNA because why? I don't see any extra oxygens. There's 2 of them, and you have AT and CG. I don't see any uracil. So in order for me to copy genetic material from the DNA, I have to open it up. I have to break down or open up the helix. Hence, I use this enzyme called helicase. It's going to unzip. You can see here, it looks like a zipper. It's going to unzip the DNA into 2 strands. You have the 5' to the 3'. We already saw this, right? And then the 3' to the 5'. This strand up top here is called the leading strand, and the strand in the bottom is called the lagging strand. Okay? Now, 5' to 3', we're going to talk about how it's easier to use because it's going in the right direction. We call it the lagging strand because it takes, when something is lagging, it takes longer. It takes longer to copy this strand than this strand, okay? So the first enzyme you need to know is helicase. I could ask you, what does helicase do? And you'll tell me it is the enzyme that unzippers the double helix to make DNA single-stranded. And we already talked about, this is replication fork, or the fork in the road right here, so to speak. Now, this is what the RNA does. See, I know this is RNA because do you see this extra oxygen? And also there's a uracil. So CG, that matches up always, right? But A, the adenine, typically matches up with the thymine on DNA, but this is RNA. So the adenine has to match up with uracil. Do you think I could have this question on an exam? I could ask you, what's this? You'll tell me, oh, this is DNA because it has what? It goes ATGC in it and it has no oxygens. This has to be RNA because it has uracil in it and it has that extra oxygen right here. And what is the function of the RNA? The RNA will copy. And it likes copying from the 5' to the 3' direction, the leading direction. So it's easy. So it copies it. And once it's copied, now we have a copy of the original message, but now it's in RNA language, a language that the ribosomes can understand. Because the ribosomes don't understand DNA language, it understands RNA language. Let's get to the other picture, next picture. Okay. Now, how does it all start? Right? It all starts with RNA primase. And if you look, ASE, that means it's an enzyme, right? Enzymes break down things and speed up or catalyze reactions. So RNA primase, when you prime something, that means you're starting things up. So the RNA primase, its function is to start things up. And in order to continue it down the line, we need DNA polymerase. And poly means many. So we're trying to make a polymer, right? So DNA polymerase then helps the RNA copy everything. Now, with the leading strand, it's easy because it's going in the 5' and 3' direction. With the lagging strand, it does this weird thing because it's going backwards. So in the lagging strand, you have to have like RNA primase, it goes, it has to jump in sections. So the way it does it is, it builds these things called Okazaki fragments. So it starts up here, and since the lagging strand is in the wrong direction, the RNA primase and the DNA poly go backwards. So they jump ahead and then they go backwards. So they do this Okazaki fragments. Then they jump ahead and then go backwards. Then they jump ahead and they go backwards. So you could see why they call it the lagging strand, because it takes longer than the leading strand. because it's running in the wrong direction. So in order for me to build this chain here of RNA plus DNA, I got to jump ahead and then do this section, then jump ahead, then do this section, then jump ahead and then do this section. You can see how longer that would take. So the RNA primase and the DNA poly or DNA polymerase only can go in the 5' and 3' direction. And that's why it has to jump ahead and move backwards in pieces on the lagging strand. And that's called, and those lagging strands and those little sections or fragments are called Okazaki fragments. Doctors Mr. and Mrs. Okazaki worked on them and they got a Nobel Prize for that. And of course the DNA poly continues on the chain, but it has to jump ahead and has to go backwards because remember, the lagging strand is in the wrong direction, or not the wrong direction, but in the opposite direction. And what glues it all together or ligates it? DNA ligase. It connects all the fragments together. So as a summary, you have DNA primase. What does that do? It primes or starts up the RNA copy. You have DNA polymerase. It helps move the chain along, and it only goes into the 5' and 3' direction. You have DNA ligase, which will glue up or connect or ligate the Okazaki fragments. And you have Helicase, which unzips or creates what do you call that? A leading and lagging strand of DNA. So DNA, instead of it becoming a double helix, it becomes a single strand because of helicase. Do you think I could easily give you a situation and then give a list of ASE or enzymes? Sure, I could easily do that. So why am I giving you all my tricks and my strategies? So that you can now recognize for future reference what's important, what's not important, and more importantly, how could it be asked of you to repeat back this information. Okay, and this is an example double helix, and you see here he stated there's 10 billion nucleotides. That means there's 10 billion of these connections. That means the code is almost endless. That means the code can code for everything, right? So now, what do we now know? We now know how the code works. What does DNA and RNA look like? How does the code copy each other? How does it read the code? Now, let's talk about how. Now, you could see like on DNA polymerase, I forgot to mention regarding controlling the code. On DNA polymerase, there is a proofreading function that DNA poly uses. When you get into cell molecular biology for future stuff, you're going to know that there's more than one polymerase. But for right now, there's only one polymerase that you have to do. Do not do this cheek cell DNA extraction laboratory. But if you look at it, it's got some really neat notes that we already kind of saw in the other class, the other lecture. And you can see this DNA double helix. All it is you take all those coils and you compress them, and then you take these proteins, they're called histones, and then it helps coil it even more, coil that even more, coil, supercoil. That's actually what it's called, supercoiling. And then when it supercoils, it forms these chromosomes. And then you have 23 pairs of them. One set from your mom, one set from your dad. So overall, you have 46 chromosomes to make a whole human being, okay? And we're gonna talk about mitosis and meiosis shortly. But you could see, this should amaze you that this code, and there's billions of them, and they're all tightly packed into 46 pairs. And all this entire code can be seen or can be interpreted in every somatic cell in your body. Soma means body. So if I get a cheek cell, and in theory, I can go in its nucleus, I can extract all of this and everything that you are. And now we know that even behavior. Everyone says, "Oh, obesity and all this stuff, it's learned behavior." Yes, partially, but according to the American Psychiatric Association and their genetic studies, behavior 40% of behavior is in here. It's in this code. It's the reason why if your dad was a hothead, you're going to be a hothead. Because if your mom was a super nurturing and caring person, guess what you're going to be. So it's not it, but there's also outside factors as well that also change who you are. For example, if I am not a nurturing personality, but if I did not have my mother around, kind of forcing me to go into the health sciences, forcing me to, and yeah, it was force. When I got out of the military, I didn't want to do squat. I just wanted to just fool around and play Nintendo all day. But she did a wonderful, marvelous thing, kicked me out of the house and told me, you need to go to something medical, anything, CNA, medical assistant, home health aide, anything. I chose medical assisting because it was fast, easy, and cheap. Well, it goes, gee, I'd go anyway, so, but it didn't matter. But I just wanted fast, get it over with, and get back into my house. Little did I know that all of that exposed me to all of these things, and that's the outside world, right? Then it goes, then it would enhance whatever, that was in your DNA as well. Now, what else is also in my DNA? Hypertension, alcoholism, and diabetes. So what do I have to do now on the outside world? What did I do? I quit drinking because alcoholism runs rampant in the Garais side of my family. Diabetes runs rampant on the Garais side of my father's side. So I have to be I try to be as healthy as I possibly can. I go to the gym almost religiously, right? Because why? Because you can control the external, and guess what happens to the DNA for the future generations if you now start changing things with you? It will start changing. And classic examples, my children. None of my children drink. I have two adult children. They don't drink whatsoever. I tried to, when they were younger, when they were teenagers, tried to make them drink. They're just simply not into it. And you could see how very different. They grew around a lot of alcohol because of my family, but they didn't take, because not only because of nature versus nurture, not only because of DNA and genetics, right? The Chinese have an interesting proverb. It probably sounds cooler in Chinese. As a parent, if you don't take care of your demons, the demons will then be passed on to your children. And the same can be said for behaviors or learned behaviors. And I'm a big fan of learned behaviors for diabetes, because when you think about it, who taught you how to eat? Your parents, right? So yes, there is a diabetic component in DNA, but I also believe there's also a very strong component in the way you were raised and the way you were taught how to eat. Another classic example is my two eldest. For whatever reason, they drink Coke, but not a lot. But my children, especially my older children, they don't like juice that isn't watered down. They think that if straight up orange juice or straight up cranberry juice, they think it's too strong. They water it down 50%. They can't stand it, and it's because why? Ever since they were babies, what did I do? For a multitude of reasons. I wanted to decrease their sugar, but also, as a parent, I wanted to make that one bottle of cranberry juice last way more than a week. Just being cheap and trying to make ends meet. But what did it create? It created children who now have a different behavior and different DNA because of the way they were raised, right? So nature versus nurture, both of them affect each other. Your nature, which is the DNA side, and the nurture, which is the behavioral and how you grew up and what and what you surround yourself with. So knowing that, we have the nice little psychiatric slash psychology connection there, because it's really important to know and understand that your behaviors and psychiatry has a strong place in this world, especially with the 256 million people on antidepressants. That behaviors, it goes, and the way you think and the way you feel, that's all chemistry. And we actually saw a little bit about that when we were talking about the endocrine function and cortisol. So let's see, let's go back, because we also have to talk about how does this all replicate, mitosis versus meiosis. And there's this lovely video here that the Khan Academy has. Okay, let's watch that together. Let's see if it will allow for sound. I think I disabled my sound somehow. Oh well, I'll just talk you through it. Okay, there's two types of replication regarding cells: mitosis and meiosis. So let's get that picture out here. Now let's go over mitosis first. We already mentioned that a whole human being has 46 chromosomes, or 1N, right? N means just number of genetic material. So you have 1N from daddy, 1N from mommy, which will give you a total of 46 chromosomes. And now we know what chromosomes are. They're just tightly packed DNA in supercoils. So 1N is worth 23 chromosomes and 2N is worth 46 chromosomes. So in mitosis, let's move the picture down here. The overall goal in mitosis is exact daughter cells or copies of the original. So mitosis is utilized in somatic cells to multiply. So if you have a heart cell, right, and it has 2N genetic material and you want to make more heart cells, you want to make exact copies. So the genetic material has to double up first and then split into two, and then you'll have a 2N and a 2N. So what questions could I ask? In mitosis, what's it for? Well, it's for somatic cells if you want an exact copy. What do you start off with? Well, I start off with 46 chromosomes, also known as 2N. And what do you end up with? 46 chromosomes, also 2N. And that's how I know it's a somatic cell. That's how I know, like, if this is a skin cell, these will be skin cells. And it'll be the exact copy. If this is a heart cell, these will be heart cells. And that's mitosis. Think somatic cells, think there's only one set of division. The genetic material has to be doubled up in between here. And you may have learned that in your biology class or in high school. If you remember prophase, metaphase, anaphase, telophase, right? Those are the divisions of cell division. And you recall our last lecture, the centrioles, those two microtubule things that help divide the cell into two, right? And then equally split them to make two exact copies. And that's mitosis. Now, what's meiosis? Meiosis, just like mitosis, start us with 2N, 46 chromosomes. But it splits twice. So the 2N then split and then make what they call germ cells, 1N and 1N. And then the 1Ns then copy, double up their genetic material, and then divide again further into 1N. So you start off with 2N material and you end up with only 1N, or 23 chromosomes. Meiosis is for germ cell layers and for the creation of gametes. And what's gametes? Sperm cell in male and ovum or egg, ova, meaning one. Ovum is one, ova is many. in female. So what does meiosis create? It starts off with 2N, which is a whole cell, right? 46 chromosomes. But what do you end up with? Cells that are 1N. So it could be a sperm cell, and sperm cells only have 23 chromosomes. Or it could be a mature follicle or an egg from mommy, and that's what? 23 chromosomes. Okay? So this one diagram is the worst I could do to you. I could ask, which divides twice? Meiosis. Which start off with 46 chromosomes? Both, right? Does not look like a both A and B question. Which ends up with 2N? Mitosis. Which ends up with 1N? Meiosis. Which creates somatic cells? Mitosis. Which creates germ cell layers or gametes or sperm or egg? Meiosis. I just went over every permutation on how I could use this to ask a question. That's how you study. You look at something that was discussed that you know that's one of our goals, and remember that was one of our goals. Then you look at every permutation on how a question could be asked. You write it down and do what? And then now you're fully prepared for any exam. Our valedictorian, Grace D, I love her to death. That's all she ever did in medical school. Maybe I shared this for you guys. Stop me if you've heard this one. She used to come up with these question sets, Alpha, Bravo, and Charlie sets. Alpha sets were questions that were definitely going to come out. Bravo were good to know. Charlie, don't even bother looking at. That's trivia, right? It probably won't be on an exam. Probably be on a board exam, but won't be on an exam. She was almost 99% correct all the time. It was amazing. And because why? She's doing what I'm trying to teach you guys how to do. You look at your goals, and then you ask yourself what's important. Thus far, when you're looking at the DNA video that we looked apart, if you can figure out all the ACEs like helicase and all that, and primase and all that, that's one set of questions. Knowing the difference between DNA and RNA, that's another set of questions. What are Okazaki fragments? That's another set of questions. What's 5' to 3'? That's another set of questions. Mitosis versus meiosis, that's another set of questions. So that's cell growth and division. Cellular differentiate. How do you differentiate between somatic cells and germ cells, right? We also, like, how does it separate? We already know how to do that. Now let's look at protein synthesis. And I believe that's a video in your lesson. Sorry, I'm bopping around, but that's I'm going in the order of no, they don't have it. They don't have it. Let's look for it. Let's look at Google, YouTube. You can see some of the bizarre things that I typically search in: comedy, jazz, and kittens, apparently. So, protein synthesis. Let's find a nice video that's got like a lot of views, and hey, 2.5 million views. So let's look at this one. What is going on? What? My computer says I am not muted. All right. I guess I got to talk through this one as well. Now, we already know that because in order for protein synthesis to occur, the blueprint DNA that's inside the nucleus has to go outside and goes to make proteins. And the middleman we already know is RNA, right? And RNA is single-stranded and DNA is double-stranded. Okay, so let's see. This may be too. This one's a little better. So that's the cell. And we already know, look, Golgi apparatus, mitochondria, you have your a rough endoplasmic reticulum, and now we're going into the nucleus. We already know what chromosomes are. And then if I loosen all of them up, forms chromatin. You have your histone proteins here and your coiled DNA. This is a really cool video. Maybe I'll put this up. And they form genes, or the genetic code of who you are. Genotype is the code. Phenotype is the expression or the physical expression of this code, okay? And we already kind of know that. So what happens, right? I think we skipped over. First, we have, of course, helicase, which opens it up. Then RNA poly, after RNA primase. And then what does it do? It starts copying. We already know this. And how do I know this is RNA? It's single-stranded, and it has uracil in it. This is part of the double-strand DNA. And the 5' to 3' is going to go smoothly, which is the leading strand. The 3' to 5' is going to take a little bit longer. So the RNA, and specifically messenger RNA or mRNA, right, this is going to make copies. And that copy is the message. Hence, they're called messenger. Now, that messenger RNA then goes out of the nucleus. It floats along those little highways, those microtubules that we talked about. And then, remember those dots? This is ribosome. You have an alpha and you have a beta subgroup, and they go together like Lego. And the ribosome then starts reading the messenger RNA material. When it reads it, you see that T-shaped, looks like a cross. Okay, everyone see that? This is what they call transfer RNA. So what is the transfer RNA going to do? It's going to match up to the code and then now transfer an amino acid on to the ribosome. The ribosome then forms peptide bonds in between these amino acids, and these amino acids are in a specific sequence. So it's a factory, and this is their factory worker that's putting together the chain of pearls or the polymer known as a protein, and it's in a specific code. And then it, because of the bonds and all that, it forms specific shapes. So let's replay what we already know, right? We already know the enzymes to unzip this and how it does it. Let's kind of move this a little bit. Let me, yes, right? We already know the enzymes that does this, right? And the formation of messenger RNA. Messenger RNA then sneaks out of the cell via those protein channels. And well, here's the picture of it, which is really neat. Because remember we had the bilipid layer? So does the nucleus, and it has certain holes. And these holes have trap doors and open and close. So if it recognizes that what messenger RNA is, it'll open up the door, and you have more messenger RNA in the inside than you have in the outside. So it will then do what? Through osmosis, it's going to go through and go out. Then it hooks up with a ribosome. You have the alpha and beta subunits. They get together. It starts reading the messenger RNA. Then it takes the transfer RNA and then transfers the genetic code to a specific amino acid in a specific order. Okay? And then once it has the specific order, the ribosome makes proteins. And all proteins are chains of amino acids linked together by a peptide bond. This is happening to you every second of your life. As long as you keep on taking in oxygen and glucose, and now you could see glucose then gets broken down to ribose, and that's what fuels and functions for your DNA and RNA, which is crazy important for your survival. So now we know how the code is read, how it's copied, how it's controlled. Now we know the structure. Let's now look at some Punnett squares. Oh, this is the one where the link doesn't work, but you can look it up. Let's look it up. So what's a Punnett square? I gotta find out why I have. Oh, I think I know why I have no sound. No, my Bluetooth isn't on. All right, I'll find out later. Okay, so what's a Punnett square? It's these things, and we kind of saw that a little bit when we're talking about things like sickle cell anemia and how traits are passed on to people, and how some people are carriers and other people have full-blown disease, and how other people don't have the disease whatsoever. So when you're looking, let's look at this biology video. Now, the alleles, remember those chromosomes that we showed you before? Each one of those little leaves of that little like 4-leaf clover, those are called alleles. And on these alleles, right, this is how a Punnett square works. Ugh. Why did they remove it? Let me just look up. Okay, let's look at Punnett square example, sickle cell. Something that's not so complicated, that's very simple. That's not simple. This is simple. Okay. So we already know about this: nucleons, chromosomes, and alleles are the part of these little leaflets on your chromosome, and we already know that chromosome is just all coiled up DNA. So let's look at this. So it goes, sickle cell is an autosomal recessive disease, meaning that if you have 2 of the same recessive genes, you're going to have full-blown sickle cell disease. Now, these letters here, all of these letters represent genes or alleles or DNA. Those are called genotypes. And phenotypes, as mentioned earlier in this lecture, are the physical manifestations of your genes. So for example, if you have a genotype for brown eyes, your phenotype would be, of course, brown eyes. If you don't have, if your genotype has absence of color in your eyes, you will have blue eyes, right? So genotype are the letters, phenotype are the physical attributes that the letters represent. Now, you could be, right, homozygous dominant. Homo means the same. Dominant means 2 capital letters. You could be heterozygous dominant, meaning the dominant, capital A, right, will show up versus the recessive S. Or you could be homozygous recessive, which you have 2 recessive genes or these two S's. So what does that mean? If you have AA, homozygous dominant, you're going to be normal. There's no, you'll have no disease. If you're heterozygous dominant, right, the dominant trait is no disease, but you'll be a carrier. So these two people are carriers. And if you're homozygous recessive, so you have the two recessive genes, this person's going to have the full-blown disease. So if you take two heterozygous people, they look normal on the outside, but they're carriers. If you, this guy marries this girl right here, and they're both carriers, or they both have sickle cell trait, right, which is heterozygous dominant, they're both not sick, or they just have some minor signs and symptoms. They have children. 25% or 1 out of 4 is going to be perfectly normal. 2 out of 4 is going to have the trait, or 50%. 1 out of 4 or another 25% will have full-blown sickle cell disease. And this is just one example of inheritance. And this type of example of inheritance, especially sickle cell, is called autosomal recessive, because it is the homozygous recessive gene that gets expressed as the full-blown disease. The heterozygous dominant gene expresses as carrier status, and homozygous dominant, the two capital A's here, right? That person is normal, has no disease. Okay. So I could ask a question about this. How does this form? And how does this form? A and A form capital A, capital A. A and S combine to capital A, capital S. It goes S and A form A and S. S and S, do you see how it matches up? And that's how that's the very, very basics of a Punnett square. That's the easiest way I could show you. And the classic example is sickle cell anemia, which it can be, for a person with sickle cell disease, very, very debilitating. I used to have to give morphine to some of my patients. That's how much pain they're in. And think about it. What does sickle cell affect? It affects the anatomy of the red blood cell. The function of the red blood cell is to carry things. If the red blood cell can no longer carry oxygen, carbon dioxide, and glucose very well, what's going to happen? My patient is not going to process oxygen very well, so it's like depleting them of oxygen. So you get very, very crampy. Have any of you ever tried to run without warming up? Like maybe a pit bull was chasing you or something like that, right? I had some bizarre examples this morning. You'll cramp up really fast. Why? Not enough oxygen getting to your muscles, increase of lactic acid, and then you'll go cramp up and it'll start to hurt. Now imagine you have that just by doing nothing, and that's your sickle cell patient, and that's some rough, rough stuff. So it is at this point of the show that I'm gonna start. If anyone has any questions regarding the